as my knowledge is concerned. So yeah, you will find actually he was very much concerned about ourselves, right? I mean, what kind of topic he wants to deliver, right? Because sir told me everyone is talking about computer vision and all. So let me think about something very different. So then he asked me, so Deepak, would it be better to discuss something about neuroscience, how computer vision is related to neuroscience? Then I found also it is very useful. So it will be definitely helpful for all of you. So sir is also huge experience, right? He has worked with several universities outside India and all, and he's currently also active. He's currently working as associate editor in the respected IEEE transaction on image processing and pattern recognition letter. He's also associated with several top tier conferences such as CVIP is conducted in India, and several other conferences also happening outside like ICPR and all. So with this introduction, I would like to request Professor Anand Chaudhary sir to start the session, sir. Over to you, sir.
Thank you, Professor Deepak, for your kind introduction. And we just keep trying. Means so he has said a lot of kind words, but my point is like we just keep trying, and we fail. But then even if we fail, we should keep trying, and that is I suggest everybody to do. So yeah, with that note, like what already Professor Deepak has said, like I felt like like we should tell something which is probably aligned with the theme of this FDP, which is modern computer vision. So there was one student in our group who has been working, on for his PhD in close collaboration with Institute of Neuroscience here in Kolkata. So I felt like this could be an interesting topic, and that's why the title of my topic is computer vision for neuroscience. And even straight away I start with an acknowledgement, because at Institute of Neuroscience my collaborator is from the Department of Neuroradiology, Dr. Mona Tiwari. And I was discussing with her that what would be an interesting and appropriate title, because initially I was thinking of neuroradiology, but then in my talk you will see that we will also talk about whole slide images or histopathology images, so which is a little bit beyond radiology. So this title actually was suggested by her, computer vision for neuroscience, and I immediately accepted. So let's start. We have this plan. So I'll have a very few slides on computer vision, because we have been listening on this computer vision for past few weeks in this beautiful FTP through the experience of many learned speakers. So I'll spend only a few slides on it, and then I'll try to show a connect, because I can talk about one of the problems in neuroscience, and which are currently being addressed by computer vision. And as you can see, there are three problems, and I'll mostly focus basically on brain tumors. And so there are three different aspects of it. Like, so one is segmentation, another is classification, and another is like biomarkers or genetic markers for prediction of tumors. So these three problems are related to brain tumors, but they are about different aspects of it. And then I will have these conclusions, and instead of saying it as a future work, I'll show some recent trends, like some of the works going on around the world, um, maybe 2024, 2025. And again, like if you just Google, it's a huge area, vast area, a lot of people contributing. So this will give you like, or this will open some windows in front of you, and then you can go back and start exploring more and do some fantastic work. So let's start with introductory computer vision. And again, this is not really a definition, but if you look at this website, which is the website of British Machine Vision Association, so they just say like what is or what falls within the scope of computer vision. So it is concerned with the automatic extraction, analysis, and understanding. So that's a very strong word, like understanding of useful information, and that actually covers a lot of things. So the input could be a single image, could be a sequence of images, often it could be a video, And like it has this theoretical and algorithmic basis, so very strong theoretical and algorithmic basis to achieve automatic visual understanding. So again, you see, like if you look at the last few words, like the very important words, visual understanding, right? Like so from I think like many of you have probably gone through this Gonzalez Wood's beautiful book on image processing, and there is in different chapters they have these different sayings, and in one chapter I remember still like an image is thousand words. So it has a lot of information. So we want to have like visual understanding and try to do it in an automated manner. But however, I would say definitely for at least like medical imaging, we should be cautious and we don't want to fully automate. We always have an expert in the loop and then try to assist him or her. So numerous application areas like healthcare, surveillance, biometrics, geoscience, remote sensing, robotics, and these are again just a handful. There are lots and lots of applications in our daily lives, in research, like where computer vision is being applied. Now, let me just show you. This is an interesting picture where the difficulty level of the task gets increased or enhanced if you go from left to right. So on the left, like you can see, it's a question of classification that what you see there is GAT or not. Now, if you look at the second picture from the left, then you have a task which is in addition to classification. So you also localize. You don't only say where I mean like whether the image contains a cat, but then if yes, like where in the image lies the cat. So that's called a localization problem, where you have this bounding box. That's often standard way of detecting or showing some object. So it's a single object. Go to the right hand side, like so maybe the picture which is like the second last from right, where you have three different species like cat, dog, duck. So it's an object segmentation, or you can also say semantic segmentation. But then you have bounding box for each of these animals. And then finally you have instance segmentations where you, even if there are two cats, for example, like you discriminate between them. So that's the most difficult one, like the instance segmentation. Now, as you can see, this is very standard computer vision, and interestingly, you will have these similar mappings. Like when you go to the medical domain, like we see this problems appear in medical domains, and in today's focus, like problems related to neuroscience as well. This is a second task, which as you can see, even if you have like any smartphone, like you can check, like when you take a picture, like it shows really where the faces are. So this is like a face recognition um, problem, and you can see there's multiple faces, and the computer can catch us, the smartphone can catch us like multiple faces. Well, there could be some interesting problems, like so maybe if you have multiple faces, then the question comes like which is the most salient face and so on. So there are issues with relative saliency and so on and so forth. But anyway, like this is a straight face detection. But even if there are multiple faces, one should be able to detect it successfully. So, for example, if you see here, like this is a tilted face, like, and then it's kind of getting missed. So it looks like that if the faces are in frontal positions, then it's easier to detect. So still there are a lot of interesting research going on in this face detection. Well, so with just this very short background, like let's try to connect neuroscience. So typical problems in neuroscience and which are addressed through computer vision. So before going to that, let me talk about like some imaging and signaling modalities. So some are not necessarily image, but they are also pretty common in neuroscience or neuroimaging. For example, we have this CT computed tomography, MRIs, and very popular techniques. I mean, popular imaging modalities. Sorry, not techniques, or you can say techniques to acquire information. Functional magnetic resonance imaging. Then you have DTI, diffusion tensor imaging. So these are pretty much, I think, within the radiology, but then you can also have whole slide imaging, as I said, for which I changed the title of my talk from neuroradiology to neuroscience, because in this talk you will see a lot of applications of this whole slide imaging or histopathology images. And then you can also definitely have like signals, like for example, electroencephalogram, like EEG signals, and so on. So this is again some problems in neuroscience. So related to neurological disorders, like so we have Alzheimer's, Parkinson's, multiple sclerosis, stroke, bipolar disorders. So this is like how, and we will see like in the next slide. So what is the connection with computer vision? So this slide is independent of computer vision. It just tells you about like different example problems in neuroscience. So some could be related to tumors, like the focus of today's talk, and these are different types of tumors: glioblastoma, astrocytoma, oligodendroglioma. Then neuroscience also deals with structures and tissues within brain. So hippocampus, as you can see, responsible for memory and learning. Cerebellum, responsible for neurodegenerative diseases like this AD and Alzheimer's, Parkinson's, and then you can have like brain cells where you want to have like detect brain cells for different. That is called the cytoarchitecture. So you can gather information about different cells. Then you have like the subcellular structures, like so even within a cell, like from microscope images, you can detect like mitochondria, or you can get involved in detecting tissues like gray matter and white matter. So there's a lot of interesting things going on in the brain, like which are of interest to us. So here is the computer vision connection, because just like few slides back, you have seen the segmentation, and you have seen like also instant segmentation, and see some examples down the line, probably next few slides. So like if you look at the segmentation, which is still a bulk of the motivation in computer vision, like so we have semantic segmentation. So that means like you decide like so I just mentioned the structure mitochondria. So whether it's mitochondria or background, um, or let's say in the next work like hippocampus or background. So it's like whether this or that. But in case of instant segmentation, you have this individual entities. So for example, like if you are focusing on brain cells, then the task is to detect this individual brain cells, not only like just saying that whether this is a cell or not. So it's a I think a more challenging problem, because you have to discriminate between individual cells. Remember that picture which I have shown, like where you have two cats. So the task doesn't end in saying that this is a cat or not, but then if there are two cats, like you have to discriminate between two cats. So the other aspect of computer vision, which is also associated with machine learning, is classification. So, for example, different types of tumors, like so, whether it's like a, oligodendroglioma or astrocytoma, and so on and so forth. For disease classification, so whether it's a normal control, whether it's like a, early impairment, late impairment, or like Alzheimer. So, in our group, we have been studying like these problems, like as you can see, like on papers which are appearing in the slide. And then disease staging is related to classification, but it's a slightly different problem. So, for example, let's say in case of Parkinson, you say here, but it can be still, like mentioned within the broad umbrella of classification. But you say they're like, okay, so at what stage of Parkinson the patient is? Like, so all are Parkinson patient, or maybe like you have stage 0, which is a normal control. But then if you have like stage 1, stage 2, stage 3, they denote the severity of the disease, and then more accurately detect. I think it's useful for the community. It's like, of course, for the doctors, and then it's helpful for patients in preparing therapies and so on. Another type of problem is prediction. And again, you see like these segmentation, classification, prediction. So these type of problems are very general problems. And these problems like have also a lot of implications outside the healthcare domain. So I'm just talking about neuroscience or focusing about neuroscience, but at the same time, you should think that these are very general problems. For example, like one student in our group, so he's pursuing a problem like let's say the anticipation of actions, action anticipation. So that's also prediction, right? So in this case, like the three problems I will discuss, they are actually all related to brain tumors. And an example which I gave here is like genetic markers or biomarkers, popularly known as genetic markers, and how they are responsible for brain tumors. So this is a work which was published in very recently, actually, transactions, computational biology and bioinformatics. Some of the papers above, for example, the mitochondria versus background. That's a work with Arctic University of Norway, and that got published in graph-based representations in pattern recognition, also in 2025. For hippocampus versus background, it's a popular conference for image processing, IC 2023, and so on and so forth. So let's look at some, examples, interesting examples, before I really delve deep into the three problems, like where I want to furnish more details. So this is an example of semantic segmentation, and as you can see, we have this axial, sagittal, and coronal views of ground truth hippocampus, like a smart grade, and versus like you have, the results from proposed. This is hog means, it stands for histogram oriented gradient. So a hog-based loss function which we proposed in IC, and it pretty much matches with the ground truth. So this is an example of semantic segmentation, because we're asking the question whether it's hippocampus or not, it's hippocampus or background. In contrast to that, this is an example image from this Vadori et al.'s paper, which is in RxIV 2024. Here you can see sample patch from Missile stent to the histological images, you can say histopathological images of the brain. Now on the right hand side, what you see in different colors, different brain cells. So remember, like that cat example, right? So you don't only say that cat or not, but two different cats you have to distinguish. So here also it's not like just cell or background, but like you have to distinguish between different cells. So this is like an example of instance segmentation. This is an example of classification, and you can see the motivation. So from the diffusion tensor imaging, this was one of the works which, initial works of this rigid MyPS system, and it appeared in expert system with applications. So these are like some information which you can get from the diffusion tensor imaging. So EPI stands for ecoplanar imaging, FA stands for fractional anisotropy, and MD stands for wind diffusing. So what I want to convey here, like through this set of pictures, is like in the top row, if you look at in the three images in the top row, so they are like EPI, FA, and MD images for an Alzheimer patient. On the other hand, like in the bottom row, these are for a normal control. It's like a person who didn't have or doesn't have like Alzheimer. And you can see like noticeable difference in the images. So this is just a motivation, like why computer vision should be used for tackling problems in neuroscience. So these are some illustrations. So with that, like let us now go to the first problem. This is on brain tumor segmentation, and this was published in IEEE Engineering and Medicine and Biology Conference, EMBC 2022. And once again, like when I discuss like these three problems, like there are some more detailed intriguing mathematics, like basically like or so mathematics is kind of language of science. So when you design an algorithm, like you have to represent the algorithm. So one is like you mentioned statements, but then you also mathematically represent. So what I want to suggest you is just look for like the philosophy, and I think in short time it's difficult to really get a full grasp of really the technical details. But please feel free to ask me questions at the end, and also feel free to look at the paper, and then if you have further questions, like please email. So I think like when we focus on getting the the main aspects of the works. So tumors are abnormal groups of cells site near the brain, and as you can see, like they disrupt normal brain functions. So the goal is like can we segment, because if we have an accurate segmentation of the tumors, then that can help in diagnosis and also in surgical planning. And our task was more compounded because we want to do the segmentation in 3D. So here is an example like where you can see the demarcation of tumor margins. It's very difficult to find where exactly or how long the tumor actually protrudes, because they are diffusively infiltrated. So they are going to the brain, going to sort of going inside the brain, and it's very difficult to find the margins. So the above view is 3D, and in the second row you have like three different views, like three different 2D views. So what we have done here for this problem, like, so we looked at like classical segmentation techniques like graph cuts, level sets, um, and in fact, like we'll combine like graph cuts with deep learning here. But these classical segmentation techniques are can give you like quite accurate results. Um, but then they depend on manual seeding for graph cuts. Like you have to, mark certain portions to show the class, like whether it's a tumor or background, or like for level sets, like which is a PDE partial equation based strategy. So it depends on initialization. On the other hand, like deep learning has like very, like good representation in terms of features, but then they also often ignore domain knowledge. So the idea is like, can we combine best of both worlds, like deep and classical? In fact, that has been like one of the, like I would say like interesting, characterization of our groups. In our group, like we often try to mix really deep and classical solutions so that we can get best of both worlds. So the straightforward way to do is like, for example, what was suggested ISBA 2017 in early days of this kind of combinations is like you simply cast it. So this was shown for lung nodules. So you take the output of one and then apply it to the other. Now the question is like, can we do better, right? That's a natural question. So so these are two very different paradigms, right? Like, so can we really develop like more intricate connections between these two very different paradigms, and naturally reap the benefit of both the worlds? The other question is like, to tackle the problem in 3D. So here are the contributions. So we have this 3D segmentation framework, very popular. Like I think many of you might be knowing, like UNet, proposed by Ron and Barger. And then the graph cut, like, so you can see the timelines, right? Like, so one is 2015, 2016, the other is 2001. So I mean, you can see the time difference. So the question is like, can we combine them? And we have shown like very interesting combination. And then we use like probability maps of a voxel. So it's what is pixel in 2D is a voxel in 3D to belong to the object class, which is tumor, and the background class, which is non-tumor. And we harness information from the UNet to improve the energy function of the graph. So see, some of the terms might be new, but in next few slides, at least you will get some idea like what we mean by energy function. And also, if you look at the third bullet, like new expressions for data terms, witness term, and the weighting parameter in the energy function of the graph. So let's see like what are these. Okay, so this is just as a motivational slide, like we can very accurately get really the tumor. So you can see like 2D and 3D views of the tumors based on like the combination of deep learning and graph cut, so which we call as deep graph cut. Okay, so let's look at this famous it's a slide or famous concept from Wyckoff-Joly's paper at ICCV. It's the flagship conference on computer vision. ICCV stands for International Conference on Computer Vision. And what you see is, like two terminals, like background and object. And you see here, like this PQRWV. So these are really, different pixels in 2D or voxels in 3D. So what you see, two types of connections. So one is, so all the pixels you connect to S and T, both S and T. These are called like T-rings or terminal rings. And the other is like you connect the neighbor pixels. So for example, like if you look at, the central pixel, it connects to all of them. So these are like the neighborhood links or N links. And also you can see the, some edges are thicker than, the other edges and so on. So this actually depends, the affinity of a pixel to belong to a certain class. Now, what is expected is, so initially, like all the pixels, like, and remember, like this is a semantic segmentation question. Like, so we want to have, each pixel, a level for each pixel, whether it's tumor or not. So initially, like if you think about this, this is like object terminal, or which represents tumor, and this is background, non-tumor. So every pixel you connect to both object and background. But once the graph cut finishes execution, then you have this one of these links are like kind of severed. So you have, each pixel is either belonging to the background side or an object side. So that means you have an idea that which is object, which is background. So in this case, we constructed a 3D, weighted graph, and as you can see, each voxel is a vertex, and then we have this source and sink, and then what I just said, like you have this T-link and N-link. So this is the neighborhood links, and this is the target links. And any segmentation is a voxel-wise classification of all voxels into either object or background. So optimal segmentation would give you like the best segmentation. That means like which corresponds to minimization of an energy. So what is this energy? So this energy can be constructed, as you can see, this is the function of segmentation. So if you have a different segmentation, you have different values. So that value of segmentation for which the energy is minimum is the ideal segmentation. So you have two terms, like one is called like a data term, and the other is called like a smoothness term. And then you have like a weight parameter, like balancing these two terms. So let's look at these like Rx and Pxy. Like what are these? We will see just a little bit later on. But just like this is kind of for each pixel we construct really a data term, and then if you sum over all the pixels, then you actually get this data term for the image. And then similarly for each neighborhood links, like you have this smoothness term, as you can see, Pxy. So these expressions will come later on. And then similarly, you have this for the entire image. So unit, you might be familiar, but I'll just very quickly go through some of the points. Like, so one is like it's a fully convolutional network, so which is applied to segment image. Is very popular in medical imaging, and multiple, of course, convolution and layers. But then it excludes fully connected layers. Since it excludes fully connected layers, that's why it has less number of parameters actually as compared to other deep architectures like BGG16 and so on. So it's an encoder decoder network. Like, so we have this encoder as the contraction phase and the decoder as an expansion phase. So if you look at this, so here like this reduces spatial dimension, and then these are some details of the architecture, and then the synthesis or expansion is recovers actually its object details and the dimensions. And here are some more details, like for example, we do max pooling to achieve, it's variant to translation and so on. But the main thing is like the expansion contraction structure. And this is important, as I have said, like since it's a fully connected actually. So in fact, like this is interesting, like it has fully convolutional, sorry, it's a fully connected, fully convolutional link. So it excludes fully connected layers, and that's why it has like less number of parameters as compared to Galaxy VGT16. And it works based on like weighted cross entropy loss. This is popular. So what we do is like we look at now this R and B, these are like the deep learned data terms, like for object, which is tumor and background. And these are the probabilities, like X is object, and C events comes from the unit. So that means using the information from the deep network, which is unit in this case, you derive expressions for the data term in the background. Similarly, like you have for the background, and again, certain information from the, as you can see, these expressions for delta xy, these are coming from, so this probability x equal to object unit, probability y equal to object unit. So these are like different voxels, and the difference belong to the background and difference to belong to the object. So without going into mathematical details, what is important to know is, or important to note, is we have a first unit, and then from the unit, like based on or without going for direct unit-based classification, so you will get a probability map which voxel to belong to the tumor and the background, and use that information as input to GraphNets, and then finally define your decision. So this is the weighting parameter. Again, there are ways. So the weighting parameter is necessary in general because, so one way to go is like you have a fixed value of the weighting parameter for each pixel into B or each voxel in 3. But on the other hand, like depending upon the position of the voxel, like which is whether near the boundary or it's far away, you can have an adaptive version of this weighting parameter, so which we have done here. So again, like based on some analysis, you can infer that whether you want to give more weightage for that particular pixel to the data term or to the smoothness term. So this is, as you can see, if you compare this expression with, the previous expression where you just have graph cut, now you have like deep graph cut. So the structure of the equation remains same, except that now we have like new expressions for this R and P. So this is the algorithm here. Like, so you compute like the data term, the smoothness term, and the weight factor, and then like the final energy function for graph cut. So here are some details about the data set which we have used. Like, so this was 2000, I think we published in 22, so it's a BRATS 2018. At that time was popular. Then you have training validation split, and more details are there. Also for the implementation, and we use like this dissimilarity coefficient for quantitative evaluation. So ablation study actually helps you understand that how your solution works, or like how really different components of your solution contribute to the overall solution. So it's a very standard technique of actually evaluating your solution, because sometimes like we might put more things into the pipeline just to make it more attractive, but actually like we are then performing some redundant things. It's not mandatory, or it's not necessary that all the components of the solution will influence the solution to the same extent, but nevertheless like they should be showing their values. Means like they should show some contributions. So here what we have done is like we compared like this is graph cut, and then with different, so this is a standard work of the graph cut, but then if we have this A is like just the data term, like you use the deep graph cut, just the smoothness term, use the deep graph cut. And then you use like all the or if you use like just like the this data term and the variable weight factor from deep graph cut. But if you use all three, then you get the best result. So that means, remember that we we used, or we harnessed the information from the output probability map of unit designing, or in modifying different components of the graph cut energy function. So all the components are showing their part. In the second case, like what happens is if you use just 3D unit, and if you use like deep learning graph cut the way as a simple cascade, not really exploring these intricacies we have done, then you can see like there are improvements, but not to the level we got. And then this is reflected like visually, as the deep graph cut matches also visually maximum this ground truth, GT's ground truth, as compared to like the other algorithms. And here are more experimental details, as we are able to outperform a lot of existing techniques. So we had this one first problem like where we use deep graph cut, and then we obtained like, so we actually combine like deep learning, classical computer vision, and then we obtain competitive performance. So the second problem is on brain tumor classification. So here, this was published in the ICPR in 2022. It's a little bit later than the previous one in ENBC. So here the task is to look at a course to find classification of gliomas. So gliomas are like the primarily malignant tumors, and here it shows that if we use two different modalities. So for example, like in MRI, so MRI can tell you that some information. We'll see like soon, like some course information, but then it doesn't tell you like the entire story. So on the other hand, like if you have histopathology, then these glial cells exhibit like certain morphological features which cannot be identified by MRI. So the point is like if you use multiple modalities, and nowadays this is like very common to involve like more imaging modalities, and to harness their complementary information, and then have a better decision. So combine MRI and WSI. So that's really one important take-home message. So MRI is used, as I just said, like the course classification for inferring glioblastoma. So it's glioblastoma or not. But if it's not, then like just based on MRI, you cannot infer more. So there we have this WSI, which is a whole slide image, and that can be used for a fine classification for inferring about other types of tumors like astrocytoma and oligodendroglioma. And we have this deep learning, and we see also the graph convolutional networks coming into picture. So these are certain contributions, like we have CNN, GCN, so combinations and so on. So the coarse classification. So we have this Fox CNN model, and that's used for coarse classification. Again, just for like equation, as you can see, that if you look into the leftmost picture, that tells you a glioblastoma, which is different from astrocytoma and oligodendroglioma. But then it's very diffused in astrocytoma and oligodendroglioma. So just using these images, it's very difficult to say whether this is, well, this just appears in right hub and this left hub, but could have been both in the right hub or in the left hub. So that's not a point for demarcation. But this kind of diffused structure, it's very difficult to say whether it's astrocytoma or oligodendroglioma. But as compared to that, like this glioblastoma is kind of more localized, and you should be able to tell using the MRI. On the other hand, like for the histopathological images, as you can see here, like A is normal, and you can see like the glioblastoma and astrocytoma and oligodendroglioma, they appear quite different in this histopathology images. And like especially like our task was to differentiate between astrocytoma and oligodendroglioma. So if you use different magnification levels from WSI, which is whole slide imaging or histopathology images, then you should be able to also have this fine classification. And as you can see, like in this case, we have these different magnification levels. So this is local magnification and this is global magnification. But you can see like that the things are looking as different. So that's important. So it's always important to look at the images to first get a feel, as I also showed, like in case of, let's say, classification from TTI images, that okay, so these images actually contain information which tells that I mean, we can do a good inference. So here also, like I had these two images very similar, exactly similar, then it's very difficult. Well, there should be some demarcation, but computer vision's task is also to have, even if like they are very similar looking, like can we extract information in a more judicious way? So that's also there. But I mean, here, like I'm still motivating you that by looking at different images for different problems, you get a feel that okay, this problem can be solved in this way. Again, I'll skip these details, but it's about like how you extract information from these patches. But these are the details that there in our ICQR paper. So the next is, once you have the features extracted, then you build a graph. This is a graph embedding. So you have these nodes and like the ages, and then you build a graph. So the question is, and this is for finer classification, because for broader classification, maybe I show you maybe this one. Yeah. So if you look at this, like you can actually see the pipeline. Maybe this workflow I should have shown earlier. So one is like you have Vox CNN, which is applied on MRI, as you can see, radiology, right? So if it's glioblastoma, it's yes, then you are done. It's yes. If it's no, then you have this like information extraction from histopathology images, like global information, local information, you concatenate, and then you apply graph convolutional network to infer whether it's astrocytoma or oligodendroglioma, or even if it's normal. So then for graph convolutional network, you have to do some certain things. And the first important thing for graph convolutional network is graph building. So the vertices are identified, as you can see, that with M nodes, like so these are basically like you can say like the patch elements, which are like the voxels. But then what you want to do is, and you can see like each node is represented by also the concatenated feature, like the global and local. But then what would be the connection? So what would be the ages, right? So the way you can find the ages or construct the ages, can have a bearing effect on your final solution. So here we did not actually, contribute in terms of maybe the new type of GCN or graph convolutional network, but by building the graph in a more informative way. So one is feature similarity and the other is graph topology. Again, I'll try to give you the philosophy without going into the mathematical details. So basically, in the feature space, like if two nodes they are quite close, so it computes some distance, maybe an RBF type of kernel. So then if they're close, then you kind of connect. Otherwise, like you don't connect. So that's really the feature space. Now, for the graph topology, it's more difficult because here what we want to know is we have some of the this nodes are labeled and some are unlabeled. So you have actually like three types of connections. So what are these three types of connections? So one is like you have two labeled nodes. You have connection between two labeled nodes. Well, you of course can connect if they belong to the same class. So as you can see, the classes we are looking at here are normal, oligodendroglioma, and astrocyte. So if you have to level, you pick two leveled nodes and then you see they're having same classes, then you connect. So that's the easiest part. How about the other one? When you connect like an unlabeled node to a level, so there you look at some kind of probability of this node to belong to this level node, and then if that's affinity or probability is relatively high above a threshold, then you connect. On the other hand, if you have two unlevel nodes, right? So then you connect if both of them have similar affinities to belong to the same class. So what you do for this case is, which is the most difficult case because you have less information, but still you can compute the affinity of these nodes to belong to different classes, and you look for the differences, right? So if there's different, because you don't know at this point like which class it would belong to because both are unlabeled, right? But then let's say like for these three different classes, as you can see, 1, 2, and 3, the difference in affinities are less for all three. That means whichever class they would go, right? Like they are strongly kind of sort of like attracted towards that class. So then you connect like and if both satisfies like feature similarity and graph topology, then only like you have this adjacency matrix. So one factor coming from the uh, graph topology and one factor coming from the feature similarity. Then you have we construct like a graph convolutional network and look at the Laplacian of the graph, and these are like some nice mathematics. Like so you look at like the symmetric version of the normalized graph Laplacian, so that used to perform better than the standard graph Laplacian. And then you do some basic layer-wise operation. The motivation behind using graph convolutional network is that they can, they are computationally less expensive first, and they can also be fantastic results with less amount of level data. So here, like the problem at hand, we felt that GCN could be a good choice. And then you do a layer-wise update, and then finally you have to like, take a decision. As you can see, finally you have the softmax layer, which is the best class. So we have the cast class probabilities, but then finally you need to have softmax, so that would give you the best class. And this we already actually discussed. So again, more details on the data sets, and you have more details on the implementation. Let's look at the results. So again, as you can see, now you are familiar with ablation studies, at least you have some idea. So if we use only MRI, and based on that try to take decision, only WSI, whole slide images, take a decision versus like combine both the modalities. The second ablation is, that if you use feature similarity only, graph topology only, and so then what is the change? And then finally, some comparisons with state-of-the-art practice. So if you think about the discussion here, like the take-home messages, so we have this multi-class for the classification, and then we have this Vox CNN for MRI, applied to MRI to have force classification, and then you have like GCN for fine classification, but GCN is applied on histopathology data. So the third and last problem, like which was recently accepted in IEEE transactions on computational biology and bioinformatics. So there we predicted like how multiple genetic markers can help you in detecting a tumor. So it's basically like this. Well, I should not say detecting a tumor possibly means. So how the multiple biomarkers play a role for a breakthrough. That might be a better way to represent it. So the motivation again, like the gliomas, and because like that's most common. So the marked difference, or where probably I think our method is very useful is there are studies like which deals with individual biomarkers rather than like multiple biomarkers. So this you can say is probably a first study of its kind, like which deals with multiple biomarkers. So how they simultaneously, get involved and influence like a tumor. So what we do here is we actually design a composite loss function. So this composite loss function has different components, and this is like interesting mathematical model. So there are literatures like in biology, like and or let's say like medical domain, and we discussed this with our collaborator like Dr. Kuwari, that it shows the literature shows that like if the biomarkers individually influence tumors, but then also they influence pairwise. How, for example, like if one biomarker is present, the other might be present. If one particular biomarker is present, it's likely that there is another biomarker which is absent, and then they can influence together. So the idea is, can we have a social model of, and this is very interesting. Like say, like multiple people go to a party, right? And then they can have individual characteristics. You can always have a pairwise behavior, like how a pair behaves, and then like a group of people, how they. And so exactly we take these kind of examples and try to model the behavior of this genetic markers. So these are some of the popular genetic markers. Of course, there are more, but what we discussed is like this, IDH, 1p19q, ATRXM, GMT, and TART. These are actually very popular biomarkers, and there are nomenclatures like its value is 1. This means they're mutated, for example, and value 0 wild type, co-deleted, non-co-deleted, and so on. So these have like popular, like I'd say like the standard sort of meaning. And let's look at like tumor type. So we have glioblastoma, oligodendroglioma, astrocytoma, and you see what we have just seen earlier, right? So we have this IDH wild type, no 1p19q correlation, PTRX wild type, TART mutated, MGMT methylation. So if you go back, so you can actually look at these different combinations which are popular for glioblastoma. And this is different from oligodendroglioma and different from astrocytoma. So we are trying to look at like how the markers basically can influence this tumor. And we use WSI, whole slide images. And remember, like, so this is basically biological evidence. And we try to develop a computational model for this biological evidence. So this is a part of pre-processing from the whole slide images, and then you have different magnification levels. How you select patches, like so we extract more important patches, like which has more information. Then this is the workflow, and we'll talk about the components of the loss function. But as you can see here, that we have these patches of local magnification. Then we have, so these are extracted from whole slide images, and then you have image patches. Then you have CNN, you have extracted features, but then you have like 3 different losses, right? So you have this MLWCD loss, conditional probability loss, and spectral graph loss. So we'll talk about these 3 losses, and this is really then naturally back propagate when our system. So let's look at 3 different losses a little bit critically, even if we don't understand the mathematical details. So first is individual contribution from the genetic markers, and here just we used a weighted cross entropy loss. The weights are added to this standard entropy, cross entropy loss. So the weights are a probability of presence of biomarker, a particular biomarker, and probability of absence of a particular biomarker. And then you can sum over like the number of biomarkers to get, this individual contribution or weighted cross entropies. Then you have this pairwise contribution. So this is far more interesting and far more non-trivial. For example, presence or absence of, so this is the general information, presence or absence of one biomarker influences the presence or absence of another biomarker. So here is an example, and this is the biological literature. So TART, like which we have seen, TART mutations, they commonly occurs with 1p19q co-deletions. So that means if this is there, so the conditional probability that TART mutations are there, then like it's highly likely that 1p19q co-deletions should also be there. So this is framed as a general structure, like as you can see here. So P of I given tau, sorry, P of I equal to tau given j equal to psi. Predicted probability of biomarker I equal to tau given biomarker j equal to psi. And similarly, like actual, and then you can have the actual probability. So we have actual probability and predicted probability. The difference will come in the loss function. So we choose 5 biologically relevant relations. And as you can see, I just like to remind you, if you go back to this slide, you can see that, so what does this 1 and 0 mean? So these are characterizations of these different biomarkers. And that's why you see these are popular expressions like P of tau equal to 1 given h equal to 1 minus tau equal to 1 given h equal to 1 and so on and so forth. So you have this different like okay, so this is like the one is predicted, one is like reference. So you have some reference values. Based on that, you have this information and you want to minimize the loss between predicted and reference. That's why you see like there is a R for reference and this is like predicted. And similarly, like now, once you go beyond like the pairwise information, so we use like conditional probability to model, this pairwise information, and then you have like group-wise information. Again, you can see International Journal of Molecular Science. So, I mean, you can see like here we have a glioblastoma, and you have different. So, okay, so IDH wild type present, no 1p19q correlation. So multiple, markers are present, which is, difficult to model using pairwise behavior. So there we constructed a graph. It's a complete graph. That means, so the vertices of the graph are basically like, these markers, and then you have ages. So every vertex is connected with every other vertex, makes it a complete graph. But then the age weights are different. So as you can see, the age weights are 1 and 1 by square and so on. Again, without going into the details. So we vary really the characterization, of these age weights. And so for different like markers, like you have, from the biological information, different age weights. So again, how you really bring in, because remember, like this is a deep network and you have to compute or you have to formulate a loss function to cover that. So we have done that for weighted cross entropy loss, like single biomarker. We have done that for pairwise loss for like, 2 biomarkers. And this is for collective, influence. So here, like we look at 2 graphs. One is for the the reference vector from the reference vector. One is from the predicted vector, and then we actually construct their Laplacians, because the graph Laplacians and the eigenvalues of the graph Laplacians, which are these lambdas, they actually capture like more information. So if 2 graphs are very similar, like so their eigenspectra, like spectra of the it's like the eigenvalue distributions of the Laplacian matrices would be closer. So what we want to do is like for a reference vector, like if we have a graph, right? So and for the predicted vector, we have a graph. So we look at the eigen difference and we try to minimize it. So now eventually we have these 3 components, and then we try to have this experimentally chosen, and then we try to minimize it. And then again, like ablation study. So if drop one loss, what happens? Like, and if we drop like another component of the loss, what happens? And if we keep all, naturally, this is best. And we have a lot of state-of-the-art comparisons as well. See, many of the works, I would say, like works on just single biomarkers, some on 2 biomarkers, very few on 5 biomarkers, but we are doing pretty well over there. So these are the data sets. In fact, like we also actually curated one data set, because from the histopathology image and also the biomarker information, we kind of collated so that this is now available in our paper, so people can use it for this kind of experiments. It's an implementation. So we have predicted actually like 5 genetic markers from WSI data, and as I said, like actually incorporated social behavior for individual pairwise and wise traits. And as I just mentioned a few minutes back, that we have created a benchmark data set and so on. So conclusions, like we have addressed 3 problems, all related to brain tumors in neuroscience, and we showed like how computer vision can be applied in tackling these problems. So we had segmentation, classification, and prediction. In segmentation, we had combination of deep and classical models as 3D unit and graph cuts. For classification, we have a GCN CNN combination, as you can see, Vox CNN for MRI on MRI for post classification, and GCN on histopathology for fine classification, and then finally for prediction of multiple biomarkers for brain tumors and segmentation of other important structures in brain. Uh, sorry, so this is a basically this should be somewhere else. Like this is, please ignore this. But like this prediction is further improved classification by genetic marker data. So some recent trends, as I said, like I didn't want to say like the future word. So like I think this is very important, like explainable AI, specifically medical imaging. We have been also exploring this in our group. We had a PRL paper like where one person like worked on this explainable AI in having the Parkinson disease classification. So this is a paper in by initiator at ISPI. And then developing human connectome project. This is very interesting. Like so neonatal cortical surface, like if there are any deformations at very early stage, like this can be uh, addressed. So this is done in like so human connectome project has been there for like from 2010. Now this is a developing human connectome project. It's kind of an add-on to that where people look at neonatal cases. Radiogenomics, we'll see a lot of omics, multi-omics. So here, like AI predicts tumor mutations and molecular markers from imaging. So this is appeared in Journal of Big Data. As you can see, a lot of papers are from 2024 and 2025, and then multimodal deep learning combined several modalities: fMRI, TTI, MRI, PET. These some of the modalities we have seen at the very beginning for this talk, and then you can have more accurate predictions. So with that, like I end before I end, like I have a lot of acknowledgement. So this is Arijit was phenomenal in for his PhD in working on this. It was in turn like work for the TCBD problem. So he also did very well on it through. So I thank like Dr. Mona Tiwari. She was instrumental. We had a lot of discussions, which based on which like Origins PhD progressed. And then Dr. Risan, like he was earlier at Padova at South Bank, so he helped on one of the. He was involved in one of the papers at TNBC, and he was presented the work. For funding, I acknowledge my university for infrastructure and internal funds, and I also thank TCS. They have a wonderful research scholar program, like for funding Origin and also enabling him to present his work at IC. So with that, like thank you very much. And for more information, like please feel free to visit my homepage and our group page, and do drop me emails like that is.chaudhary@university.in. Thank you. Okay, so already I think um, there are questions. Yes. So should I take questions? No, I think it's time.
Yes, sir. Sir, you can.
Okay, so okay, let's see. So that was first question was this is the second question. Like I think let's before that there was the first question. Okay, so the first question was, sir, can we use GCN for Fisher fusion in multimodal architecture? Yeah, that's I think good question. Well, I mean, you see, like for when my scholars or even let's say masters undergrad students they come like and ask like a research question, so then I say like I don't know the answer because that is what we are actually exploring. So I feel that's very interesting. So if you look at GCN actually, like so the GCN, I mean, if you model a problem, so this is I think this is kind of a summary I would say. So you have to see whether you can cast this problem as a node leveling problem or as a graph level, as a graph classification problem, or as an age classification. So you should look into the GCN architecture, and I'm sure like GCN has a lot of potential. There are a lot of variants. In fact, we also work with some variants of GCN, like a dynamic GCN. Like there was one paper appeared in SM transactions, like from a group at Stanford, and we use like the idea for our GBR work. So for multi-feature fusion, and I think like if you want to have like less layered architecture, because in GCN we don't have like a lot of layers, like a few layers. And then the interesting part, I think, like one of the interesting aspects is also the graph building. So for sure, like I think you can use this. Okay. Now the second question is, can multi-scale feature fusion help capture both fine grain tumor ages and global brain context? Yeah, again, like we used it for still for brain tumor classification. um, but I mean, you are right actually. Like, so the reason, like, we use both or let's say like different degrees of magnification, or one we say like a global feature or local feature, like to have more information integrated in the feature vector, and then after that, like, we kind of build a graph. So the answer is yes, like you can certainly use like multi-scale feature fusion and to capture like more information. And yes, like, so fine grain you can think of like local, and so global brain context is like it needs to be clarified, like what do you exactly mean. But generally speaking, yes, like, so you can actually integrate like types of uh, different type of information by looking at different magnification. Any other question? Okay, so, and how explainable AI methods highlight which imaging biomarkers are most influential in brain tumor prediction? Okay, so here actually we did separately apply explainable AI methods. Now, what happened in this case, specifically for this biomarker prediction problem, is we have actually a lot of biological evidence. So biologically, like, we could explain, like, these tumors are responsible. Sorry, these biomarkers are responsible for these tumors. Maybe like if multiple of these markers are present or in this form, like they cause like this tumor. So, but we didn't have a model of computation. So here actually, like, the evidence was always already there, and then we try to use like this computational model, like where we look at the individual genetic behavior, pairwise behavior, and collective behavior. So, I mean, generally, like when you have, let's say, a segmentation problem, like uh, generally, like explainable AI is very important. Like, when you try to, let's say, segment, classify, because you can say, okay, like, because of this happens, and so on. In prediction, it might be, applied, but here, we try to model the existing biological information. So could be, like, explainable AI. We haven't explored it really. I mean, for this problem really, when I haven't thought about it actually, because I was, or we were happy actually, like in sort of like developing computational models for biological information, what was already there. But yeah, it might be good, like, to look at like explainable AI in this case. In fact, like, there are nice explainable AI models available for CNN based architectures, but for GNN, or like TCN is one of them, we still don't have a lot of explainable AI. So the explainable AI for GCN, I think that's also one area like where a lot of work can be done. But coming back to your question, we haven't tried, but it might be possible. We have to think about it.
Yeah. Any other queries from the participants? I don't see any other questions. So, sir, we will wait for only 1 minute. Maybe they are
Ah,
typing.
sure, sure, sure.
Yeah.
So you can see, like, it's a very interdisciplinary area.
Yes, yes, sir. Yes, sir. Yes.
Yeah,
I mean, anyone can also pick up this kind of problem statement, which is
yeah,
really
sure,
challenging and all.
sure, yeah. But it will be always, I suggest, like, it will be always good to have some people from medical domain in the team.
Absolutely, sir. To guide us.
Yeah, exactly, because I think like this also happened for my PhD, like so the problem should come from them actually, and then we look for answering them. Because see,
Absolutely.
like there is one way of doing medical imaging, like okay, maybe like there are available data sets, and then you look at like certain methods and you develop your own method and try to outperform the existing methods. Well, I mean, I will not say that I have never done it. Maybe one or two cases I have done it, but I think more we should understand like the utility or the requirement. Then I think more we should approach actually like people from other domains, and that would make it like a very good collaboration.
Absolutely, sir. Yeah, so participants, I think there are no other queries. Let us thank our speaker, Professor Anand S. Chaudhary, sir, for giving his valuable time to us and sharing his knowledge how computer vision can be applied to neuroscience.
Sure.
Thank you, sir, on behalf of Computer Science and Engineering and the E&ICT Academy at NIT Jaipur. Would like to thank once again for accepting our request and delivering a wonderful talk on computer
Thank,
visions. So
thank
we'll be happy to have you in person, maybe in some other events in
you.
future.
Sure, sure, sure.
Yeah. Thank you. Thank you
Okay,
so much, sir.
thank you. All the best. Bye.
Thank you, sir.
Okay, so I could leave, right?
Yes, sir. Thank you.
Thank you. Thank you.
Yeah. So we'll be moving on to the next session. The next session will be delivered by Professor Devi Prasad Dogra from IIT Bhubaneswar. Will be speaking